I'm Nadim Ajami. I'm assistant professor at the Molecular Virology and Microbiology department at Baylor College of Medicine. It's nice to see that the room is kind of split between bioinformatics and biologists, with the exception of Nicola and Morgan and some others. I can tell you that we've had ads out in our center for years looking for good computational biologists. So I'm pleased to see that there's a congregation of you guys here. I'm more on the biology side of things, but I've tried to merge a bit on the bioinformatic dark world. So usually Joe Petrosino, our center's director, is the one giving the talks. I'm usually more in the lab making sure that all samples come through and data comes out at the other end. But since he's probably touring the world somehow, somewhere, so this is me. So this is Texas, this is Houston. This is the Texas Medical Center. It makes a good splash. In the background, you can see downtown Houston. This is part of Baylor College of Medicine. It's not our building, but it's a previous building, so that's why it gets the title slide. That's actually where the Human Genome Sequencing Center lives, in the top 3 floors of the Alkek Center. I've lived in Houston for the past probably 10 years, and I've never stood where this picture was taken, so it might be Photoshop. This is our newly built McNair Hospital. Baylor decided a few years ago that it needed its own hospital to continue the training of medical students instead of having them pass through the Methodist and other hospitals in the medical center. So we have a very nice and interesting microbiome study going on there. We had the opportunity to sample the facility before it opened, and we continue to sample the facility as it opens. So it's a nice story. Unfortunately, I won't be talking about that today, but that's it. So our center is called the Center for Metagenomics and Microbiome Research. Our mission is to understand how microbiome impacts health and disease, and to ultimately translate these findings for the production of better therapeutics, diagnostics. And first and foremost, actually, is to serve as a hub for these activities internationally. We want to do this by enriching established studies and develop new projects. We have a pilot project funding system in place. We also want to develop means to study host microbiome interactions by some basic science that goes in the lab, and at the same time develop and recruit critical mass for system biology approaches to microbiome research, hence my ad for computational biologists. At the end, the goal to translate the findings to the clinic. Since we are in the medical center, it's heavily driven by physicians, so we have the opportunity to take what we develop in the lab to the bedside. If this is our mission slide, then this will be our vision slide. Right now, we are at the early stages of this wheel. We are dealing with microbiome explorations by looking at host and microbe interactions. We're starting to build models of our microbial ecology and later therapeutic development. We want to get involved in policy outreach. This is one of the topics in microbiome research that it's in its infancy. There's nothing written about it, and we want to be at the forefront of that. At the same time, we have an education component. We are part of Baylor College of Medicine, so we are developing curriculum to specifically deal with microbiome courses. As I mentioned before, we want to translate all our findings, or the ones that could be translatable, into the clinic. We also have a commercialization arm. Now it's called Diversigen. It used to be called Metanome. Some of you in this room have worked with us through the company. But this was pretty much built to satisfy the demand of big pharma getting into, and small biotech getting into the microbiome space. Since we're part of the medical center, I just want to give you a brief introduction. It's not really history, but it's our environment. So there's close to 50 institutions all across the medical center. There's 26 agencies of the government. There's 26 private non-for-profit health-related institutions. In terms of patients, we have over 7 million visits per year. There's close to 100,000 employees, 34,000 full-time students. I think UT is a little bit larger than that. In terms of clinical impact, we have close to 7,000 beds, and every time I present this slide, I conclude by saying what I get from all of this is that we have millions of samples available. And these samples come from your usual suspects, your gastroenterologists, your infectious disease doctors. But often enough, you get a cosmetologist with a freezer full of stool samples. You don't know why, but comes to the lab, say, I want to do microbiome research. And you say, come, go ahead. Part of our collaborations involve diseases in diverse body sites. You'll recognize some of the institutions here. Of note to this group, we work with the Forsyth Institute in the oral microbiome. We are doing complete genome sequencing and assembly and annotation in over 100 oral genomes. We have a very large, our flagship study with University of South Florida looking at diabetes and the determinants of type 1 diabetes in the young. We collaborate a lot with MD Anderson, as you might imagine. Some other institutions in the Northeast. In terms of bioinformatic analysis, we collaborate with Curtis Lab, with Rob Knights, and others at the Broad. We don't forget about those diseases that cannot be studied in humans, so we partner with collaborators to study them in animal models of human disease. We are a highly collaborative center. There's a few projects that are born in the lab, but we do serve as a microbiome go-to lab for anything that involves experimental design to data analysis. What we want to do at the end is it's just to model all the data that we get and see how it's contributing to human health and disease. And what we have envisioned is that by using standardized protocols for sample collection, sample storage, processing, sequencing, and analysis, we might be able, in the near future, to fuse data from different cohorts where you see common exposures and common genetic risks and identify unique regional factors such as diet, race, ethnicity, access to healthcare, environment, and so many others, and see what's common about them to increase our power to get meaningful data out of the microbiome. The way we do this is by applying the metagenomic approach. We have in-house validated sample collection techniques and kits. We've worked closely with leaders in the field that are developing these kits that most of you use, like MoBio, DNA Genotech, to validate those collection methods. We take samples from many different environments. We have samples from agriculture, from sewage, from water, rivers, and so forth. Human, we have human samples, and we have industrial samples as well. We try to tailor or to improve the extraction methods to enrich for microbial biomass, given that most of the time the host or the environment that you're studying brings in a lot of noise. So we rely heavily on automated platforms. We currently have 5 Hamilton Start and Starlet systems dedicated only for extraction. These systems can extract 200 samples every 90 minutes, and we have the flexibility of loading 4 plates on each platform at a time. So this is how we cope with the high volume that we see in the lab. In terms of sequencing platform, we have some flexibility here. We have built-in in-house sequencing capability, and we also partner with the Human Genome Sequencing Center that has a lot more sequencers than we do. In total, we have 3 MiSeqs, 2 regular MiSeqs, and 1 DX, which is the FDA-approved MiSeq. This is more for the company side of things. And then we have a HiSeq 2000 and 2500 that we use for metagenomic sequencing. In addition, we have a PacBio that we use for complete genome sequencing. PGM 454 that it's alive, quote unquote. We're keeping it alive for one project that started many years ago with that platform, and we haven't been able to switch. In terms of analysis, we have developed many tools. My bioinformatics team always tells me that they're not going to reinvent the wheel, but if they see that they can improve it, they will. So we've developed methods in-house, but we also use many of the methods I'm pretty sure you guys use. Some of you in this room have pioneered. The current pipelines that we offer to our collaborators include 16S V4 RNA gene profiling. We are at the last stages of validating the V3, V4 kit with Illumina, and we are working on the V5, V6 protocol as well. Currently also offer the ITS2 and 18S gene profiling pipelines for microbial eukaryotes, and we are again at the end stages of validating the ITS1 pipeline. We do whole genome shotgun sequencing for automated library prep. Our cutoff is 10 nanograms. Anything below that would have to be passed through manual library prep, but we've been able to do it. I'm a virologist from training, so I really push for the virome analysis, and we're able to detect DNA, RNA viruses, including bacteriophages and eukaryotic viruses. We're starting to deal more with microbial RNA-seq. As mentioned earlier, we think that we've gotten good enough to the point that we've identified, we can name the bacteria, but we need to deal more into the functional capability or potential of the microbiome. Complete genome sequencing is currently achieved through PacBio sequencing, and we're also looking at Illumina Maypair libraries right now to see how well we can produce drafts of those genomes. In terms of qPCR, a lot of our collaborators excuse me, are interested in quantitative data, so we've set up the qPCR looking at V8, V9 regions of the 16S gene, and we are working on a V4 to hopefully be able to use this data and compare it to the 16S gene profiling. We also have culturing abilities and capabilities in the lab. We do aerobic and anaerobic culturing. This slide presents our growth since 2011 when the center was born, and you can see that it's quite a bit of work. We have 231 projects as of March of this year with 210 collaborators, and they, as I told you before, they range in the scope. To support all this, we have a team of 26 people in the lab, including a few grad students and postdocs, but most of us are in the staff scientist route with a heavy team doing extraction and bioinformatics. 231 collaborations, of which the majority are within the states. We have 160 in Texas because everything is bigger in Texas. 79 just at Baylor College of Medicine. Currently we have 12 CMMR-born projects. They include wet and dry lab. 185 collaborators, I think that's more like 210 right now, and across 74 institutions. This is the breakdown of our current projects. Most of it, close to 70%, is still 16S. More whole genome shotgun, it's coming our way after we've developed or produced preliminary data using 16S. Almost like 5 to 10% are virome nowadays. We're getting more interest now in microbial eukaryotes, so we're seeing a lot of 18S and ITS. And as I said before, we are trying to get more into RNA-seq. How things start in our lab. So this is usually one of the slides that I present to collaborators when they come to us looking for help in the microbiome space. I usually tell them, if you have a clear question, we'll probably get you a clear answer. If you want to walk into a dark room with a shotgun, then that's the type of data you're going to get at the end. So it's always good to start with a hypothesis. Samples get submitted to the lab. Metadata gets submitted at the same time. We usually review the metadata just to make sure that every field is completed and we don't have incongruencies or discrepancies. So after the metadata is okay, we start processing samples through any of the pipelines that are tied to the project. We have processing QC, and then we start the interaction or the interface with the collaborator, where we provide them with descriptive indices, which are pretty much QC values or QC metrics of their run or their data. And then we get into an iterative process with the collaborators where we exchange information and provide them with descriptive analysis that usually results in having more questions than answers. So that's why this is a loop. You can see how this process could be endless for a non-microbiome researcher. But the idea is that we will provide the expertise, knowledge for the things that we can, and for those that we cannot, we will identify key collaborators to partner with. By no means I will expect you to read this slide, but it's just giving you an overview of the different stages of data analysis that we have in the lab. Again, descriptive indices, the value is just QC. We give them metrics for the metadata, the run QC stats, alpha beta diversity, taxonomic summaries. All of this is done independently of the metadata. So it's pretty much what you got out of the sequencing run. And then the descriptive analysis incorporate metadata, and you get values for microbial richness and evenness, and then pairwise comparisons, group comparisons, PCR plots, all the nice things we see on papers. This has, at this point, it has no predictive value, and it's just a summary of what we saw. Then one of the last stages is association analysis. We just recently hired a biostatistician to help develop this pipeline in the lab. This also contains metadata, and the idea is to build models using the microbiome data that we produce, which will result in statistical and predictive value. So what we've seen since I joined the center a few years ago is a paradigm shift. Originally, people came to us with, again, a stool full of freezer full of stool. Most of the studies were retrospective. They were pilot and exploratory. I don't know what I have, but I have 1,000 samples. I want to see what the microbiome is doing. Maybe there's a failed treatment somehow, and you suspect that it's due to a difference in microbial ecology within each individual. So all these questions come to our lab pretty much off-targeted. So maybe samples were not collected thinking about the microbiome. They were not stored appropriately. They were, they've been thought many times, but still people want to get the most data out of it. So, being a collaborative center, we said, okay, we'll look at it. These are the caveats of the study, and we look at it. More often than not, studies were not powered to find differences in the microbiome. But what we've seen lately is that more prospective microbiome studies are taking place. We've been included since the inception of the project. Collaborators come to us and say, I have a clinical trial, I want to design this project. I'm looking at the responses to chemotherapy in cancer patients. I want to see what that is doing to the microbiome. Should I give antibiotics to my cancer patients before starting chemo or not? So all these great questions, people are starting to include us when they are designing their studies. And that usually results in better data, better analysis, and more significant results. Most often now they're hypothesis driven, and they they deal with experimental and treatment design. They want to get into the mechanism of the microbiome. So again, not only who's there and what are the names of the bacteria that we find, but what is really the function of the microbiome, what role the microbiome is playing. And then we try actually to power them for outcomes. And this is one of the things that I think it'll be valuable to discuss during these 2 days, since a lot of you in the audience are biostatisticians or computational biologists, because every NIH grant is asking us to come up with study designs and sample size calculation and effect sizes. But I don't think we have a great idea right now of which metric to use, what to look for when you're dealing with just microbiome data as a whole. I want to give you a few examples of preclinical and clinical research that we're currently taking under in the lab. I want to present studies to you that will be published very soon, but and yeah, that's just the disclaimer. So one of the things that is going on in the medical center that has been having high impact in patients is microbiome transplantation, or FMT, specifically for recurrent C. diff infection. Right now, the FDA is not requiring an IND for FMT for specifically this type of disease. For all others, it is. So the goal is to investigate the comparative efficacy of fresh, frozen, or lyophilized fecal microbiota from healthy donors given to recipients with recurrent CDI. And the reason is because we do not know what works. We know that in animal models, when you give live bacteria, you can ameliorate colitis. When you give bacterial supernatants, you can also have the same effect. Sometimes even if you give dead bacteria, you might end up with a benefit. So just trying to answer those questions. And from a clinical perspective, trying to see what the differences are between fresh, frozen, and lyophilized. So quickly in one slide, I can tell you that we have our donors here with good diversity in their microbiome. And then we have our patients before fecal microbiome transplant. And then we have patients with samples collected 7 days, 14 days, 30 days post-fecal microbiome transplant. At the time that this analysis was done, we had 21 patients. Right now we have over 63 patients, and we're following them up to 56 days. So what we see is that before microbiome transplant, the community, the structural community of those recipients were pretty, they were low in diversity and pretty much similar to each other. However, after transplantation, they all shifted and they looked like their donors. I'm pretty sure you've seen the data before, but just wanted to give you an example of kind of clinical studies that are going on in the lab. What we've seen, looking at this further, I apologize, I only have the phyla taxa here, not the genus. We're completing this study with whole genome shotgun sequencing to get down to the species, but what we see is that patients that went into a successful treatment, which were 91% of them, had a bloom in verrucomicrobia, namely Akkermansia mucinifila, very early after microbiome transplantation, compared to the ones that did not. Of course, the amount of patients in this failure group is lower than the one in success, but we are hopeful with the new data we can have better power for this comparison. We also saw a similar trend with actinobacteria. Another interesting study, which is in this case is preclinical, it's looking at a mouse model for neurodevelopmental biology and determining social behavior and cognition in the context of diet during pregnancy. So we had a set of dams that were fed a high-fat diet, and we looked at the microbiomes of the offspring. We did not look at the microbiome of the dams, of the moms. So what we saw is, and there were 3 types of diets. We had a high-fat diet, we had a regular diet, and then a transition diet, which was first high-fat, followed by regular diet. So what we saw is that the microbiomes of those pups that came from moms or dams that were fed high-fat diets were really different from the ones that were in the regular diet or the transition diet. And interestingly enough, when you measure the cognitive capability or CFC memory scores, those that were fed a high-fat diet, they scored lower significantly. Of interest, these 2 blue points that are that are from mice that came from moms that were fed regular diets were the ones that scored the lowest on the memory score. We've complemented this analysis with social behavior, and the data is even stronger. I want to spend a few minutes talking about our flagship project. It's called TEDdy, the Environmental Determinants of Type 1 Diabetes in the Young. It's a study by the University of South Florida. It's a multi-institutional international setting where kids are followed up from 3 months of age until the onset of type 1 diabetes. So to date is the largest clinical microbiome study. We're producing over 40 terabytes of raw data, and you can imagine that this provides a scenario for a lot of opportunities, but at the same time a lot of challenges. Our primary goals are to identify viral candidates that might trigger type 1D, as there's evidence out there that it might be an enterovirus, or there might be an enterovirus infection associated with the onset of autoimmunity. So we're doing viral metagenomics, included virus target extraction to look at this. We also have a culture-based viral amplification arm to enrich viruses, trying to catch lightning in a bottle in that sense. At the same time, we're doing 16S, ITS, and whole genome shotgun on all samples. We have close to 20,000 samples in this project. We're collaborating with Rick Lloyd, which is a virologist in our center, and the Broad and Rob Knight's lab in terms of data analysis. So this is the how the study designed or how the study was designed. The entire cohort it's composed by almost 8,500 subjects. We have a microbiome 101 case control study that involves 802, of which we've collected 13,000 stool samples and 6,000 plasma samples, and we're looking at, again, for bacteria, fungi, viruses, both native and cultured. We're doing this by looking at the ITS, 16S, whole genome shotgun, and virome analysis. So far, what we've seen is these are preliminary results. We see an increase in bacterial diversity in stool preceding autoimmunity or islet autoimmunity onset and decrease at the time of diagnosis. So we see a bump just right before the onset of autoimmunity. In terms of microbial composition, we see we see two big groups of people when we do unweighted unifrac analysis, and we see that there's no real correlation or clustering by islet autoimmunity. However, we see that there's a clear correlation of change of microbiome structure by age, as you might expect. Your bifidobacterium is very high early in age. Your Clostridia replaces that bifidobacterium as the subjects progress. I also mentioned that we see this distinctive two clusters, and you can imagine that we as a lab spent months and months and months trying to determine what was causing that differences. We got very paranoid, and we ended up producing maps of each well in a thermocycler just to make sure that we didn't have differences in temperature and so forth. However, what we've seen so far is that this has been driven by very few tags, that you only see it on unweighted unifrac analysis. You lose that clustering using weighted unifrac. Also, when you look non-phylogenetic methods to create this dissimilarity or distant matrices, you lose that type of clustering. However, what is very interesting is that we see kids that are both just present in the red cluster or the blue cluster, and we see kids that jump from one to the other. However, the majority of the kids that are in the blue cluster stay in the blue cluster, but the kids that are in the red cluster end up jumping to the blue cluster, and that's a factor of age as well. So we're looking at the acceleration of the changes in the microbiome and what is actually driving those changes, to try to associate them with phenotypes. To follow up the beta diversity analysis, we are seeing in the preliminary data, starting from baseline, we're seeing a split or a difference in the microbiome structure between cases and controls. Controls tend to go and become more diverse over time, and while cases conserve a more similar microbiome compared to their baseline. In terms of taxa, we identify that there's significant differences in species and abundance between IA in cases and controls, with the controls having more of the good bacteria early in life, such as bifidobacteria. Not to forget our viral pipeline. We've run a comparative analysis between primary and cultural virome, and I'm showing you here those results and the ones that overlap. We've identified many different viruses down to the genus level. The ones labeled with a P are plant viruses, the ones with an A are animal viruses. Of interest, we've seen that the biggest or the most prevalent viruses that we identify in our samples are mast adenoviruses and enteroviruses, which is not very surprising, given that these viruses are very prevalent in the community and we expect to see them early in life. But looking a little further into this, we want to try to extract more information out of those sequences and see what are the reads that we can use to differentiate or assign taxonomy based on at least the enterovirus genus. Historically, the capsid genes have been used to classify these viruses, so we want to restrict our analysis to those and build those capsids to see if we can get better at that. What's next to come for TEDI? Well, we are completing the data set. Our contract expires for TEDI-1 in August of this year. TEDI-2 is coming the way. The contract is for data generation, and analysis is following up. We want to do network analysis by establishing microbiome profiling groups. look at the metabolic potential. There's another group within TEDI that is looking at a metabolome and RNA-seq as well. And at the end, the idea is that all of us will get together in one room and try to come up with cohesive ideas there. We want to run integrative analysis, looking at bacteria, viruses, and fungi, and how those networks will exist in the environment. As I mentioned, TEDI2, starting at the end of this year, we are getting nasopharyngeal samples and PBMCs as well to try to complete the story. I'd like to spend the last few minutes talking about our arm for research and development. Since we produce a lot of data for many, many projects in the lab, the bioinformatics team are working very hard to develop what we call PUMA, which is the pipeline for Unifier microbiome analysis. The idea is that we will be able to produce, or we are able to produce data of multiple different types. Data will have to be pre-processed accordingly, since they are not all the same. And then we will align those reads to different databases that we have access to, hoping at the end that we come up with a standard output where we can plot multi-dimensional data and get a good story out of it. In terms of enabling our collaborators and others in the group to run their own initial analysis, you can imagine that with 200 plus projects and 4 project managers, we don't have time to take project from A to Z without getting input directly from our collaborators. So we've designed a microbiome explorer, which is yet another one out there. And the idea is that we produce, or we enable the collaborators and others in the lab, including our students, to produce initial plots to look at their data, and those include alpha diversity analyses, beta diversity analysis, hierarchical clustering, taxonomic abundances, and summaries, and so forth. One of the coolest things that we have in the lab going on in terms of R&D is using microfluidics to concentrate and isolate bacteria from complex samples, at the same time getting rid of host DNA. We recently hired a biomedical engineer that used to postdoc in our lab, and he's now staff or research associate in the lab, and he's really leading this part. So it's the microfluidic approach is based on DEP, which is dielectrophoresis. It's a very, very old concept. It's by no means a new field, but the novelty of it is that it's being applied to bacteria I think in the right way. So it's a microfluidic device that has an inlet. It's dielectrophoresis, so it's analogous to your gel electrophoresis chamber. However, the charges here are constant. The idea is that you can flow anything from 10 microliters to milliliters of sample through it, and when the depth field is on, so when those electrodes are charged, you can see bacteria attached to those electrodes, and this is based on their biophysical and bioelectric properties. When the depth field is off, then you can see them floating through the chamber. I'm going to show you a quick video of how this works. So here in the top, you see a representation of how bacteria will react to the charges that we see in the chamber. And in the bottom, we have a top-down view of the chamber. You can see it's the size of the electrodes here, and the bacteria are labeled with red fluorescent protein. So bear with me this video. I'll show you that this works. Come on. There you go. So bacteria are floating. I think the flow right here is going. Yeah, it's passing through. So now DEP is on, and you can see the bacteria collecting in these electrodes, and you can see that everything will pass through. So you might imagine that you can use this to test drugs, to test metabolites, and how bacteria respond to it. We also want to use it to be able to sort bacteria. So Lorenzo, the biomedical engineer, he believes that you can differentiate rod-shaped bacteria from other types of bacteria in a sample, that you can detect at a very early stage patients that will go into sepsis and so forth by looking at their blood specimens, for example. At the end, we want to couple this with a particle counter or a flow cytometer so we can sort, and then we can do metagenomics on those samples. Another type of research and development that we have in the lab is after trying many different times to look for a LIMS system or a laboratory, I forget what it stands for, information management system. We decided together with the Clinical and Translational Research Center or Institute at Baylor to design one for us to be able to manage all the information. And I'm proud to say that last week we launched it, and we only had a few problems with it, which is totally expected. But it's been an effort that it's been brewing for the past year and a half. With that, I would like to acknowledge all the people in the lab and the Human Genome Sequencing Center, funding agencies, and take any questions.
Yeah, I'm a virologist, so I have a question on the virome.
Absolutely.
So I work with plant viruses, and the big problem is to understand which viruses we have in our samples, because we don't have the 16S and this kind of stuff. So do you also have this problem in your field? Like to understand the variability and to understand what you have there. Also in the concept of the recombination that you can have between viruses. So, for example, I work on a virus that was derived likely by reassortment because one genomic segment is really similar to viruses that infect fungi. So maybe if I do an analysis, I only find that segment, I can imagine that I found a virus that can infect fungi, but instead is a plant virus. So what do you think about this topic?
True. So I think the method that we've developed are great for first generating hypotheses and to inform subsequent analysis. Right now we run virome analysis on the 2500 using the 2x150. So you can imagine that in 150 or even 100 bases, you have limited amount of information to identify recombinations or reassortments. We also work with a lot of reassortment viruses. As far as the plant viruses that we see in these kids, my suspicion is that they are from food. There's other studies that have compared vegans and omnivores and so forth, and you can see those viruses segregate as well. So I think that this data is great to generate or to inform new methods. And if you can isolate those viruses, then that's a good way to study them. But I don't think where we are right now with this short read is going to be enough to characterize the full genomes of all the viruses. We usually run maps, as you saw for noroviruses, but the coverage that you need it's quite substantial. And we get around that by looking at longitudinal data. So we usually pair samples from the same subject across time to try to build those genomes as we go. But I don't think the data has the resolution enough to answer questions in terms of recombination or reassortment.
Thank you.
Are you working with any interomics comparisons yet, and how are you delivering that to collaborators?
So we started working with the metabolomics core at Baylor, and right now what we've received from them are flat files, and at this point we're just running correlations between abundance of different metabolites and the abundance of taxa. It's simplistic at this point. However, it's been great to generate new hypotheses and to test those hypotheses later in a basic science scenario. But we are eager to see how we can, multi-layer multi-omic data or layer multi-omic data.
Since throughput doesn't seem to be a problem for your facility, I'm curious how you've integrated sample replication into the design of some of your studies.
Yes, so that's a very important question. We definitely have high throughput capabilities. We usually run 2 MiSeqs every week. To address the question of reproducibility and contamination and so forth, we have built-in controls with every run, and those those include a negative control, which you don't expect any reads to come out of it, or a very low amount of reads depending on the sample. And we have positive controls that are based on genomes that we do not expect to see in the sample types that were loaded in the machine. We usually pair samples by their type. Whenever we have longitudinal studies that require different runs, we either collect all those samples for 16S, amplify them all together and rerun that pool several times just to come up with the amount of reads that we need. But whenever that's not possible, we try to include samples that were run previously in other runs in the new ones, just to make sure that we are not seeing a lot of noise. I'm sure you guys seen that Illumina has had problems in the past with what we call barcode ghosting, that you run a MySeq with a unique set of barcodes, then you come later with another set of samples and you end up finding barcodes that were not present in there. Since then, they've put out new washing techniques that have been able to resolve that problem. However, we keep running our own controls to make sure that's not happening.
Nadim, I was curious about your bioinformatics pipeline as someone that develops bioinformatics pipelines, and everyone has a bioinformatics pipeline, it seems. So I was wondering if, say with Puma or anything, is it publicly available and
the idea is that it will be soon. So we are working on the manuscript right now, and we are polishing up the code. It was primarily done for Teddy since we had different sample data types. The idea that the first publication will be tied to Teddy, but at the end, our my bioinformatics team is all about open source. So,
So do you hope to put it on like GitHub or something?
Yes, absolutely.
That'd be great.
With the big disclaimer that you can use it, but we are not responsible for anything that you use it for.
Of course.
Like everything else out there.
So I was really interested in your microfluidics work that you discussed, and so it looks like, of course, it's sort of the holy grail to be able to extract out bacterial communities from whole blood and do those types of analytics. But in doing so, you're going to get this enriched community for bacteria. But I'm wondering what you compare it against to ensure that you're getting the complete community, that you're really seeing everything that's there and that the approach is correct. Because when you do metagenomics on something like a blood sample, you're basically sequencing human or whatever host.
True.
And so what are your metrics for doing those types of analyses?
So right now we are the profiling stage. So what we do is take samples before and after. And right now we're based on 16S analysis to define those communities. We hope to move to whole genome shotgun or metagenomics later on. I don't have a good answer for your question, what is the gold standard right now, but we know that we are capturing 95 plus % of what we put in. The clinicians in the group say that whenever you have sepsis, you'll get anywhere from 10 to 100 bacteria. So that's a very, very low amount. Our bioengineer, our biomedical engineer, claims that he can detect that amount of bacteria. That usually involves a lot of sample pretreatment, but that's what we're working on right now. I'm sorry, I don't have a good answer of what to compare it to, but
No,
I think
it's just interesting.
those are usually low biomass, so you have to be very careful how you run your processes too.
Hey, so you talked about processing a rather impressive number of samples, and mostly you talked about sort of individual studies where you have some sort of control over the design and the technology and the interpretation. Have you attempted any adventures in comparing different studies that were done possibly using, slightly different approaches that could give you slightly different biases?
We have. So I would say 7 out of 10 collaborators ask for a control group, and they always go back to the HMP. We were part of the Human Microbiome Project, but everybody knows that that was developed using 454. The extraction methods are very comparable to what we use, but that was, again, 454 looking at the V3, V5 regions. So we know when we put V4 data and V3, V5 data on the same plot, boom, differences, right? P-values of astronomical significance. But we know the caveats of those studies. However, what we tend to do, specifically comparing cohorts with the HMP, is we use close reference OTU picking, and we slice the V4 region. So we try to come up with a data set that is the most comparable to each other as possible. Hopefully, as we build more data sets, more of our projects will have a uniform way of sample extraction processes and analysis. That way we can cross-compare better, but it's a problem in the field that everybody's using their own method, and at the end, you're putting cohorts together, and the information that you get is not very clear.
Okay, so in that case, you can't do much about the amplification bias for the different V regions.
You
You're
cannot.
trying to control the computational bias.
Correct.